almost.
Yeah. What city?
From Jonquière.
And Guillaume went straight from his PhD to working with Philips. Right? He's been working there for a few years now. He is here today to tell us about his experience working in an industry company, which I think a few of us can have questions on. So I'd like you to take a broad perspective on this. He comes from a company that does mainly imaging technology, but those of you who might be interested in other companies, say radiation oncology or processing, can certainly garner from Dr. Joubert's experience in that respect. So, thank you.
Okay, so thank you, Yves. Thank you for giving me the opportunity to present this. So I'll just get started. So I hope the sound is fine. Yeah. So I'll just get started with a very short outline of the presentation. So I will just start with a few words about me, just so that you know who I am. Yves already gave some details. And then most of my presentation will talk about research in a large industry such as Philips. I will, of course, focus on what I know, so that will be mainly medical imaging and mainly MRI. But as Yves mentioned, I believe that a lot of things can be expanded to other fields. And to show you how this is done, typically, I will guide you on the path from an idea, so if you develop something in an academic setting, to a product that would be, in the case of Philips, sold by a large company. So as you may guess, there are many different steps that need to be fulfilled before getting from the idea to the product. So I will guide you through these different steps. I have a disclaimer to do, not so much a conflict of interest. So I'm an employee of Philips, so that's probably the reason why I'm here, so you know that now. I am an MRI scientist, meaning that I only do MRI, and I will talk a lot about MRI. I know that not everyone here is doing MRI, so that I won't go into any technical detail in that presentation, but I apologize in advance if MRI is not your thing. But then I believe that a lot of things will generalize quite well to other imaging modalities or even other type of research in biomedical engineering or medical physics. I will give my personal perspective on this. So this is not the official Philips view of how this is done. This is my experience. So I've been working with Philips for almost 9 years, so I'm starting to get a good experience. So that will be my own experience. And then, if you attended the CUBIN Scientific Day, so CUBIN is the Quebec Bioimaging Network, I gave a quite similar lecture back then. So that was in February, so I apologize if you were there. I made a few updates, but that will probably look a bit familiar to you.
The audience is quite different.
Yeah, it does. I still know a few people that I know were there, so that should be fine. So who I am. So Yves already said a few words. So I'm from Jonquière, so I'm not from Montreal. I moved to Montreal about 15 years ago for my undergrad. Basically, I did my undergrad and PhD in physics at the University of Montreal. My PhD project was on MRI. So I was mainly working on vascular imaging and diffusion. My advisors were Gilles Baudouin and Gilles Soulaille, you may know them. And then right after my PhD, I got the chance to start with Philips in Montreal. So basically, I was quite lucky regarding the timing, and that's something maybe we can discuss after. But basically, Philips were back then looking for scientists in Montreal. I already knew some key persons at Philips because I got to meet them while I was a student. So connections are very important in that case. So the timing was just right for me to get started with Philips, and I'm there back since then, so I really like what I do. I also have a position of associate professor at the University of Montreal, so I remain really a Philips employee. That's Philips who pays me, but then I get to teach some courses. I can also co-direct some students, so that's basically kind of a synergy with the department there at the University of Montreal. And probably the best thing I've done is that little girl. So yeah, I'm a happy father since about 1.5 years. Yeah, so that's not the topic of my talk, but I always like to put a nice picture somewhere. So what am I doing at Philips? So the first thing that a lot of people ask is, that's quite strange. We know that Philips is not building MRI in Montreal. They have factories all around the world, but not in Montreal. What are you doing in Montreal working for Philips? So that's the question I get quite often. And I will give some details about what I do, but mainly my role is to do the interface between Philips, so an industry, and academic research. So I do not support clinical MRI. I work only in research on paper. So basically, I get to work with academic collaborators, including some here at McGill, at different institutions, to work with them on basically shared projects. So basically, very often our academic collaborators have ideas, things that they would like to develop that are also of interest to Philips. And I'm there to provide technical support, to collaborate with them on these projects. So that's my role. What I do typically is that I'm quite close, so obviously Philips builds the MR, I'm quite close to the actual data acquisition. So that's mainly what I do. So making sure that we can use the MR to the best of its capabilities and to get basically the best data out of the system. So that's probably my main task. And of course, why Philips wants to do that is that we want to remain close to the academic world because a lot of innovations come from the academic world. And we will use that input, we see what people are doing in the field to guide the Philips R&D roadmap. So basically we see in advance, or we see what is coming, what are the trends in the field. We also see what are the shortcomings of our products, so if there are things that we can improve. So by getting that feedback, that's quite important. And I do only MRI, but MRI, people do MRI, we know that it's a very large field in itself, so it's impossible to know everything about MRI. And from my position, I get to work on many different projects, so I know a little bit about most of the different topics inside MRI, but I'm jack of all trades, master of none, in the sense that I get to work on many different projects. Unfortunately, but that's life, I don't really have a very strong expertise on one specific field, which is true for a lot of academic researchers. So that's already a difference. So who am I working with? So I'm based in Montreal. So my main sites that I support are located in Montreal and around Montreal. So in the province of Quebec, that means CHUM, so University of Montreal, McGill here, Sherbrooke, so CHUS, and also in Quebec City we have the Cardiac Institute in Quebec City. And we also have other sites outside of Montreal in Canada. So I have a colleague who is based in Vancouver, so she takes care of the two sites in Vancouver, which are UBC and Simon Fraser University. But I also get to cover basically most of Canada, so that means Toronto, Thunder Bay, and also Edmonton. So we have sites doing research at quite a few different places. And basically, what am I working on? That's a very big question. I could give a lot of different answers. And the answer will very much vary depending on when you ask me the question. Basically, I get to work with many different projects, and these projects evolve over time, of course. So that's just a glimpse from the last year. So basically, ICMRM is the large MRI conference. That's the international conference of MRI. So basically, everything about MRI gets presented there. And this is basically what I was involved in the last year. So you see quite a few different topics, quite a few different sites. So that's the kind of work that I get to do. So that was about me. Now we'll switch a little bit more to how research and R&D is done at Philips, always focusing on MRI. So we have a few different ways of doing research. And development. And the difference between research and development, I'll touch upon a little bit later. So first, Philips builds MRI systems. So these systems are getting developed and built at a few different factories. So these are the factories where the actual system gets developed. So for instance, the magnet themselves, so really the magnet, comes from the state of New York. And they basically get shipped then to the Netherlands, where they will add the green coil, the RF coils, and everything. So again, and the pieces may come from all over the world. Then we also have a factory in Gainesville in the US where they are mainly focused on RF coils. And we have also factories in China where they have some systems that are more specifically for I would say the Asian market. And also in Bangalore where they mainly work on software. So a lot of the software comes from Bangalore. The headquarters, so Philips is a Dutch company. So the headquarters are in Des. Des is actually a very small village close to Eindhoven in the Netherlands. So this is where the actual product development takes place. So that's more the D of R&D, that's really the product development. In parallel to that, Philips also has a branch that is called Philips Research. So I'm part of Philips Healthcare. The development of the new systems is done by Philips Healthcare. We also have a separate branch which is called Philips Research, with doing more fundamental research. It's always with the kind of course, in the industrial setting, but it's basically looking further ahead. So things that would become product in maybe 10, 15 years. So we have a few different research labs. And one in particular in Hamburg, Germany, is working a lot on MR. So these are also Philips colleagues, but working more on fundamental or technology related projects. And they were quite successful over the years. So for those of you doing MR, there are quite a few quite important papers that came out from that research labs. So the difference is that their goal is not to create the next product version, it's to think about new technologies, new things that could become product at some point, but maybe a little bit closer to academic research, for instance. And then the third component, and I'm personally part of that component, is what we call clinical science. So on top of the scientists working on the product development, the scientists working in research, we are about also 70, 80 scientists in the field. And in the field, I mean not located at a factory, but located at different sites all around the world. And as I explained, our role is to do the interaction between Philips, the company, and our academic partners. To basically make sure that our academic partner gets what they need to do their research, and also making sure that Philips is aware of what is going on in the academic world. So I'm part of that team. We are 2 in Canada, about 12, 13 in the US, and I believe close to 80 around the world. That's only MR. So that was MR R&D at Philips, and I'll give much more details later. And then the good part of my presentation will try to explain how we start from an idea. So maybe you think about something, you think that this is the nicest thing ever, and you would want Philips to do a product with that, for instance. And I will guide you through the different steps, and I will do that mainly through examples. I think that's the best way to do it. But the general product development pipeline looks a little bit like that. And it's basically a funnel. And the first thing is that I should rename my slide something like that. So the long and tortuous path from idea to product. As you will see, when someone develops something, very often we would like everyone to use that very quickly. Unfortunately, that's very rarely the case. So it takes time to go from an idea to a final product that we can sell. But basically, it looks like a funnel. We start with input. And that input, you can see, can be many different things. It can be, of course, academic research. That's where I'm involved mainly. It can also be competition. So we have competition. So other companies are basically doing the same thing as we do. And we, of course, look at what they do. And if they come out with something that we don't have, then most likely we'll have to react to do something. And of course, feedback, business strategy. So maybe we want to be different from the competition as well. So we don't only want to do what they do, but we want to have our own flavor. So this is what drives the, so this is basically the input to the funnel. And then we have a few different phases, and the next slide will explain a little bit more these different phases. We start with what I call research. So basically that's starting from that input to then develop a prototype, so develop an idea. And that research can be done inside industry, but also inside academia through collaborations. Then once we have something that we think will work, so we have a good idea, we're pretty sure that this is something that will be interesting, will work, then we'll try to make that prototype a little bit closer to something that we could sell. So that's the 2 next steps. So advanced development, so basically making perhaps a new version of the prototype that would be more user-friendly, compatible with more systems, to get more feedback. We are also involved, and I'll explain a little bit more, but we as clinical scientists are of course very much involved in that phase as well. Because before launching a new product, the company will want to get feedback from institutions. So that's very often that we will provide access to prototypes, or in the world of MR, we often call that work in progress. We'll give access to either new coils or new sequences, and we'll want our research partners to use them, get feedback, try to solve the issues that remain. Once we have a very good idea and we have feedback from our collaborators, then we'll go into the product development. Product development is a lot about regulatory approvals. So it's really making sure then that we have a final product that we can sell. And at the very end of the funnel, you get something like a small MRs coming out of the funnel, or a new sequence or a new coil. And I'll go into a bit more details about these steps. So the first one, in the world of MR, at least, it's really academic research. So, and I feel that could be a bit different for other fields. I think in MR we are quite lucky that it's very easy to do academic research in MR, I think. One of the main things is that there is no ionizing radiation, so we can scan volunteers quite easily. So there is a very strong academic research community in MR. It's of course also true for other modalities, but I think it's partly very true for MR at least. So a lot of the innovations that will eventually become product come from academia. Some innovation don't come from academia. For instance, there are very few groups doing academic research, for instance, on new magnets. There are a few, but a lot of the innovations coming really on the hardware side, sometimes that comes only from industry. So we have our own R&D teams working on that. But new sequences, there are tons of groups doing sequence development in MR. So a lot of the ideas regarding new acquisition, new contrasts, new reconstructions come initially from academia. But the company, of course, already has a role to play there. So most of the researchers out there will be working on commercial MRs. So some groups, again, may build their own system, so that happens, but most of them will work on a commercial system. And we can make sure then that we provide them the tools to do their research. So that's also the role of the company to do that. And in MR, that means that we can give access to the post-programming environment, and we have tools to support them. So that's my role, but we have also tools like a forum, we have meetings every year at different places. So that's the role of the company to make sure that the academic research can be performed on our systems. And of course, there is a strong interest for us to do that, because if someone comes with a new sequence, it's much easier for us to use it if it's already on our platform instead of being on the platform of the competition. So there is a good incentive for the company to provide a lot of resources so that the academic researchers decide to do their work on our systems. Then the next step is what I call corporate research. So it's basically looking at what academia is doing and trying to think about it in a little bit more industrial setting. So working on the prototypes. So in academia, sometimes people are very much interested in doing proof of concepts. So they will work on something, they will show that works, I publish my paper, and that's it. I start working on something else. So that's true for a lot of different academic projects. They will want to show that it's doable, but that's basically it. Of course, that's the first step, but that's not enough to get something as a product. So corporate research will sometimes want to take what is done in academia, but make it a little bit more robust, a little bit more general, try to make it a little bit more user-friendly, for instance, so that we can get more validation done. I really starting from what is already done in academia, but trying to make it a little bit more industrial. And basically, academic research, I feel, is still very much research. So they will work on a lot of different projects. Some of them will get to product rather quickly. Some of them will get to product eventually, so in many years from now. And some of them will just simply not get to product because they will realize that this is, in the end, not so much a good idea. Or there are shortcomings that we didn't see, or we found a better way of doing that. So for many reasons, some things will never get to product. But once we have something that we are pretty sure should get to a product, then we'll go to the advanced development phase. So basically, that's where we will really optimize the implementation. So we'll make sure, for instance, we have a nice user interface. We'll make sure that calculation time is quick enough so that people will want to use it. So all these different things. And that's also where the business side of it comes in. So it's very nice to have something that we think is very good. But of course, what is important is that the people out there will think it's very good and will want to buy it. So that's the end thing. So of course, we have to look at the market. So we have to do market surveys, market assessment to see whether that is something that people will want to use. And then product development is a little bit a continuation of the advanced development, but working a lot of documentation, regulatory approval. So not so much actual scientific development or industrial development, but this is also a very important part of doing academic research, and I'll talk a bit more about that. So how do we track? So there is a pipeline, so we have ideas, and in the end we have products. How do we track the progress of one idea toward being something that we can sell? There are many different ways of doing it. The way Philips uses, and quite a few industries are using it. I know the NASA is using it. That's the one from the European Commission, is what we use, what we call technology readiness levels. So it's a scale of 1 to 9, and basically a TRL of 0 or 1 to that scale is a 1 to 9. There is also a different version which is 0 to 9. Basically, you start from a very basic idea, so that's academic research. And and then you will formalize that idea. You will develop it into something that is closer to a product. So each idea in the pipeline, so we have what we call our development roadmap, will be assigned a number that basically will tell the people where that specific technology is on the technology readiness level. And for instance, as we get closer to introducing a new feature, a new product, the product will get introduced, for instance, at level 7. So basically that's where we will, let's say that we take an example of a new MR system, we will have at least one or a few installations of that system in hospitals, where we will also track how they work. Then at 8, basically that's where we really have a product that basically anybody can purchase. But that's even not the end. So generally, the project for that product will be closed only a few years after, once we have a lot of feedbacks on the product. And we see it's always the case where you will need to do service packs or things like that to fix things that people in the field realize, but that in the industrial setting we couldn't really see. So that's the way Philips is doing it. I'm sure other companies are doing quite similar ways. And going through that pipeline, a very important thing is that we need to decide what we do. So even large companies have in the end, limited resources. So sometimes, I know that I had that view as a student that I was looking at the big companies and they seem to have money and to do everything they want. That's simply not the case. So and in the end, what will get to something useful and something that we can sell, I would say the bottleneck is never that we lack ideas. We have a lot of ideas. We have a lot of things that we know could be useful, could be nice to have. The limitation is always the resources that we have to put. So we need to decide what we are going to focus on. And this is done with maybe different ways again, but this is one example where we will put all these different items on plots like that. And the plot, basically, we have the urgency, and we have the importance of the item. The size of the dot could be, for instance, the amount of resources that we need to put into that item to make it happen. And the color in that case, so that's just an example, the color could be that these different items are associated with different projects, R&D projects, for instance. So we need to decide on what we are going to work, and so we need to really assess the importance and the urgency of all the different items. And some people could tell, yes, urgency, importance, that's more or less the same thing. So I can give just an example of how this can differ. So for instance, if one of our competitor comes out with something that they sell and they do a lot of marketing about it, we may think that in the end that's not really useful. But then most likely we will still want to have something similar, at least to be able to say, hey, we have also that. So that's something that could be urgent, even if we feel that it's not really that important. And we have the other way around, where you could have something, a very, very good idea. You know that that's going to revolutionize how you do MR, but nobody else has that idea as far as we know, and it will take 10 years to make it work. So that's something that would be very important, but not urgent. So that's how we get to define the level of importance and the level of urgency of all the different items in the pipeline. So that's my only slide about that, because that's not the most interesting topic, but it's a very important one for the industry. So in medical imaging, but that's true for all in the medical world especially. I know that in radiation therapy it's probably even worse than MR. We have to deal with a lot of rules and laws. So we have to make sure that we respect all of them. So there are basically local laws, and local approvals that we need to get. There are laws on all the product needs to be built. There are a lot of different rules that we need to follow and respect. So, and that's a very, very important part of doing industrial research, is that we have to document everything. So once we get started on something, we need to make sure to have all the documentation so that we can really show how that project was developed, how the product was built, so that we can basically answer the questions from all these different organizations. So that's a very, I would say for a scientist like myself, that's definitely not the most interesting part of my work, but it's a very important one for industry. But because if you don't get these approvals, if at some point Health Canada discovers that their product is not doing what it's supposed to do, you will run into problems. So that's very important for companies to respect. So that's a general way of how you get from an idea to a product. And what I'll do next is I'll go through a few different examples. Again, I apologize for those of you not doing MR. My example will all be MR, but I won't go into details, and I think you will be able to see how this is done in practice. So the first example I use is a relatively new technology that we call multiband imaging, or it's also called simultaneous multi-slice. So just to give you an idea of how that works, so in MR, we very often do sequences that are basically multi-slice sequences. So we will acquire the slices, excite and acquire the slices one by one to build our own 3D volume. For many of these sequences, the acquisition time will depend on the number of slices that we acquire. So if you acquire more slices, then the acquisition time will be longer. So that technology, basically, what it does is that we will excite many slices at the same time. So instead of doing the slices one by one, which is the typical way of doing it, we will excite, in that case, 2 slices, but it could be more than 2, at the same time. And then we will get 1 images from the 2 slices together. Let me look something like that. And we will use the coils, so the receiver coils, using what we call parallel imaging, to basically separate the 2 slices and reconstruct the 2 slices independently. And the goal of doing that is basically an acceleration factor. So if you can excite 2 slices at the same time, your scan will be about a factor of 2 faster. So you can see that this is very, very important in practice. So this is the technology, and I will look at how that technology started from an idea to a product. So the first idea was, we're still very much in the academic world. So that goes back to 2001. So that's already a long time ago. So that paper in MRM, they basically explained how you could do that. So basically they showed that you can excite more than 1 slice at the same time, and you can use parallel imaging to reconstruct these slices. So that's already 17 years ago, so a long time ago. The problem is that this was an idea, so their paper is a very good paper. It shows that it can be done. It doesn't mean that it's easy to do in practice. So that's very much what happened. So at that stage, what they were doing, they were exciting the 2 slices, acquiring the 2 slices. What you get is something like that, where you have basically, it's maybe hard to see, but you have 2 slices really on top of each other. Meaning that to reconstruct the 2 slices separately, it's very difficult because they are really superposed. So to tease out the signal from the 2 slices was a very difficult technical problem. So even though mathematically on paper it could be done, in practice with the coils that were available back then, that was almost impossible to do in a real setting. Maybe a factor of 2, but that's about all you could get. So basically that paper got some attention back then, but not from industry at that time. The next innovation was in 2005. So again, purely academic paper. What they showed is a technology that is called control aliasing. So they have a very nice acronym. So that group always has acronyms that are linked to drinks. So that one was Capirana. And basically, without going into details, what they showed is that by manipulating your sequence, you could move the two slices apart. So basically, instead of having two slices that are directly on top of each other, you would get something like that. And just by looking at the images, it's much easier to see the two different slices. So if our eyes can see it, mathematically, it's also much easier then to separate these two slices. So basically, it's called control aliasing. So you basically move the two slices, one with respect to the other. So that came in 2005. So that was a very important development back then. The issue was that they showed how to do that for some sequences, but not the sequences that are basically probably the most interesting for that technology. So that was already a good step in the right direction, but the industry was still not that interested back then because we still had quite a few problems to solve before making that into a useful product. I would say the breakthrough really came in 2010, so that's a conference abstract, and the paper came out in 2012, where they showed how to do such control aliasing, but specifically for the sequences that would gain the most from that acceleration. And these are single-shot sequences, so single-shot API sequences. These are used, for instance, for functional MRI, for diffusion MRI. So it's really these sequences that can really gain from such an acceleration. So these papers showed how to do that for these sequences. And that's really the breakthrough. So from that point, that becomes something really interesting because it was working with the sequence that people would like it to work with. And you will see that, so basically we started in 2001, we are now 10 years later, so that was all purely academic work. But from there, very quickly, Philips and the other companies will get involved. Actually, one of the very important developments, you may have heard of the Human Connectome project. So it's a very large project in the US. They receive a lot of money from the NIH to do that. So basically, it's looking at basically functional MRI, diffusion MRI, looking at the brain of healthy young adults. And that project, so they got a lot of coverage, news coverage. They decided to use these new sequences with acceleration to perform their project. And that project was done on the Siemens platform, so that was not done on this platform. And they came out with an implementation of that technology that was robust enough to be used in a large-scale project. So they scanned about, I think it's 1,200 subjects, so it's a very large project. They came out with still a prototype, still an academic version of the sequence, but that was robust enough, reliable enough to be used with such a large project. And very importantly, for the academic world, they decided also to share that sequence with other users. So that's the first release of the sequence that they did. So all people doing academic research on Siemens system could start using that sequence, basically. So quite quickly, a lot of sites started using that technology in the research world. So that was still not something that sold or approved for clinical use. But in the research world, quite quickly, people started to use that technology. Of course, Philips had to do something. The first implementation on the Philips platform came about 2 years later. And very interestingly, that implementation didn't come from Philips. And that's also true for Siemens. The first implementation didn't come from Siemens. That's probably an illustration that large companies tend to move relatively slowly. So small companies, startups, maybe they are more, they can react more quickly to such new trends. So the first implementation was done by a company that's called Jarotools. Basically, it's a spin-off of ETH Zurich, and they basically offer research support on the Philips platform, for instance. So they developed that version, and basically Philips users could purchase that sequence from them, but they didn't get any approval for clinical use. So it's still purely research. Then Philips itself started working on it. So the first Philips-made version of the sequence came out in 2015, and this is still research. So basically what happened back then, and I was involved in that that trial is that we distributed that sequence to about 10 different academic sites. That included some of my sites here in Canada to get feedback. So basically, before going to something that we could sell, we need to make sure that it really works. So we asked these sites, and these sites were very eager to try it to accelerate their sequences. Please try it and then give us feedback. So we did a national trial. We got feedback from these sites. Indeed, many things had to be improved or fixed in the sequence. But that's the kind of thing that we do basically for all different new products. That we need to get first a prototype out, get feedback from the academic sites to make sure that we can improve our version. So that happened in 2015. Then shortly after that, Siemens came out with their product version. So until then, people could use it on both the Siemens and Philips platform, but only for research. So at the end of 2015 at RSNA, so that's the large radiology meeting, Siemens announced their product version, so a version that would be available for clinical use. And so they announced it at the end of 2015. They got FDA, Health Canada approval later in 2016, and they could start selling that technology to users for clinical use. At about the same time, Philips also applied for FDA and Health Canada approval. Philips did a little bit the other way around. I'm not sure why, actually. First, we applied to, these are the FDA notice. So we applied before we announced the sequence, so that's quite not the typical way of doing it, but we made sure that we had all the approvals first, and then we could announce the sequence at RSNA 2016, with knowing that we already had basically the approvals for that sequence, and we could start selling it right away. And then the third large MR company, GE, also came out a little bit later. They came out with their own version of the technology. So they were maybe not as much involved before in the research world, but they of course had to come out with something. Because for people doing fMRI and DTI, that's really a breakthrough. Sequences that used to take something like 15 minutes, now you can do them in 4 or 5 minutes. So that's really, really an important development. And behind the scenes, why all the companies had to come out, and at least that's definitely true for Philips and GE, why they had to come out with implementations, is that there was another very large trial in the US that was starting, a little bit like the Human Connectome project, but that one focusing on adolescents. So that's called the ABCD trial. It's still ongoing. So basically, they will do a 10-year follow-up of, I believe it's 10,000 adolescents in the US. And that one is a multi-center, multi-vendor trial. So basically, sites working on all 3 platforms are involved. So Siemens were already quite fine because they already had implementation from the Human Connectome project that they could use. But then Philips and GE, I was involved in the Philips side. We got quite a bit of freshers from our site in the US that wanted to participate in that trial. They basically told us, you need to give us that sequence, or otherwise we'll need to buy a Siemens, because we want to be part of that trial. So that's the kind of like background pressure that the industry gets. And that sometimes we need to make sure that we have something quite quickly so that our sites, our partners, can use it. So that's an ongoing project. I guess most of you are not really working on brain development, but the data from the HCP and also from that project is available now. And that's the end result. So the end result, yeah, these look like regular DTI and fMRI sequences, but that you can acquire now much more quickly than before. So basically the factor that you see here, the NV sense factor, the multiband sense factor, that's the acceleration factor. So we can go in some cases up to a factor of 8. So that's really a very, very big difference to what we were doing before. An acceleration that has very little cost. So we will lose some SNR, but not that much. So that's really why people want to use that. So that was one example, and I would say once the breakthrough was made in 2010, 2012, it was very quickly that the industry got involved, because that was really something that was going to change how people use these sequences. So I would say a very important development. So the industry got involved quite quickly. I can give a counter-example to that. So basically that's a contrast. I wouldn't say technology, but a contrast. It's called T1 Rho. Probably you don't really know what it is, and that's not really important. what is important is that this is something that we know exists for a very long time. So that's a paper from 1975. So that's even before MRI really got started. So we were still in the MR spectroscopy days. Basically, it's a contrast mechanism. We know it's there. People have used that contrast mechanism in a lot of different research studies. So that's on PubMed. I have more than 500 papers using that type of contrast. There are some research sequences available for basically all platforms, but then still people don't really use that clinically. And what is the reason? That's really purely my personal opinion. I believe this is a sequence that is good at many things. So people have used that in the brain, for musculoskeletal, for liver, for a lot of different things. So it's good at many things, but it's not really good enough in anything. So basically it's kind of, yeah, it could be useful there, could be useful there, but I don't think there is any killer application where it's definitely the best thing we can do. So in the end, people explore that in the academic setting because that's what researchers do. They will explore how it could be used. But to be used clinically, as far as I'm concerned, I don't think there is the clear application yet where this sequence could be really that useful. So this is something that has been there for more than 40, almost 50 years, and it's still not offered as a product by any of the large MR companies. Then, what can help your idea become a product? I think one thing that is very important is consensus and standardization. And the example is probably, most of you are probably too young to remember that one. So that's one night in the 80s, so basically two different formats of videotapes, the VHS and the beta format. In the end, the VHS is the one that basically won. The beta format disappeared. But back then, there was a lot of debate. So some companies were basically betting on the beta format, some others were betting on the VHS. And that's very much a problem because large companies really are afraid to make mistakes. So if there are multiple different options of technology, they will not want to decide on bet on the wrong horse, basically. They will want to most likely wait until there is something clear that people will want to use. And that's very relevant in MRI. I took an example from Diffusion MRI. So this is an open source toolbox for diffusion post-processing. And so basically that's a tweet from that toolbox. It says that we have now more than 20 different diffusion reconstruction. That's very nice for researchers. They have a lot of tools to play with. For a company, that's very bad news because we won't implement 20 different flavors of the same thing. So we want to know which one people will really want to use. And what happens is that instead of choosing the wrong one, we just wait. So we just wait until there is a clear consensus on which one we should use. So that's very much a problem in MRI. But then I can give examples where such a consensus was obtained. So this is an example of ASL. So ASL is a perfusion sequence. So we can measure the perfusion of the brain. And basically, again, from the ISMRM, so that's a large International Society for Magnetic Resonance, the perfusion study group there, they published that paper in 2015 saying, so basically these are most of the world experts on ASL, and they wrote that consensus paper saying ASL should be done like that in clinical use. That doesn't mean that in research you can't do other things, and people still do other things in research, but if you do it clinically, as the experts, we say that you should probably do it like that. And for companies such as Philips, that's very useful, because then we know that this is what we should do, because that's what people will want to do. And again, if I look at the FDA approval for a new sequence that came out shortly after that, we refer directly to that paper in the FDA document, saying that according to the international recommendation, we do that way. So that's very useful for companies, because first we know that we decide to do the right thing, that's the thing that people will want to use, and then we can also tell the regulatory approvers that this is the way it should be done, because all the experts agree on that. So I know that's also true in radiotherapy planning, that there are some such consensus papers on how, or guidelines on how things should be done. That's something that I think is very important for industries. And I believe a last example, a very quick one, there are other ways to make sure that you have consensus, or at least that you have standardization across different vendors. That example is a different one. So that's MR elastography. So that's a technique that we use to measure the stiffness of the liver. And the stiffness of the liver will tell us the degree of fibrosis. Basically, if you purchase a MR elastography package on any vendor, you will get exactly the same thing. And basically the sequence and the post-processing will be exactly the same, and the reason is that it all comes from the same people. So this is a company that is called Resundant. It's the spin-off of the Mayo Clinic. And why are all the vendors using that sequence? The main reason is that they own very important IP. They have patents on the technology that are very key to the technology. So it was basically impossible for the different MR companies to find a way to do MRE without infringing on one of the patents. So I don't say that's the best way to do it, but that's the way to do it. If you have a very, very good idea that you're absolutely sure everyone will want to do, you could make sure that to patent it and then to sell these patents to the companies. But that's one example where that worked. In a lot of cases, it doesn't work that well, because if a company is not sure that this patent will be really important, they will just avoid using it, or they will wait until the patent is expired before using it. So in some cases that could work, but I wouldn't say that's the best way of doing it. But in their case, they basically achieve kind of a forced standardization by owning the IP for the technology. So just a very quick summary, and this is again very personal, so that's not an official Philips site or whatever. If you want to get your idea to a product, basically, I think these are guidelines that can be useful. First, you need to work on something that will solve a problem. So basically, and if you pick an important problem, that's going to be even more interesting, of course, for the company. You have to do something that people will want to use. So you may have a solution, but it could be extremely difficult to implement, extremely difficult to use, too time-consuming. Maybe that's not something that the company will want to use. As I mentioned, we live in a regulated world, especially in medical imaging. So you need to do as much validation and documentation as possible. So you need to make sure that everything in your code, in your tool, or whatever is well documented, that you know exactly what it's doing. It's always a good idea to talk to manufacturers early, because if they know you exist, if they know that you have something that is coming that could be very interesting, it's going to be on their radar. And they maybe will be more interested in working with you. If, on the other hand, you do everything quite hidden and at some point you publish your paper, that's the way of doing it in the academic world, of course, but perhaps it's going to be more difficult to get the attention of the vendors. And I would say the most important one is be patient. So in the best cases, it will take many years. In some cases, it may take forever. So yeah. That's my own opinion. If you are interested by that, there is that session from the ISMRM. So I'm not sure if you need a password to get in, or maybe someone who attended this like ISMRM could give you their password. It's basically a very good session that it talks specifically on that topic. They invited people from most MR vendors, I think all MR vendors, to give examples again and to explain how an idea or a prototype could get into a product. So if that's something that is of interest to you, I really recommend, and you have video recordings of the session. It's quite interesting. That's it. Thank you.
Very interesting talk. And even if I've heard it for the second time, I still learned something.
Okay.
Before we open up to questions, I'd like to mention that I've booked one of the meeting rooms here, and it might be useful for the students to come by and meet with Guillaume. I was thinking somewhere like at 2 o'clock. Does that work for students who'd be interested? In room 7001 here, so we'll make sure he's there. But we'll have lunch first. So we have some time for a few questions before we break up. James.
Attention, attention, code info, urgence pédiatrique, rouge 1. Attention, attention, code indigo, emergency needs,
that's even longer.
red 1.
Thanks for the talk. Was just wondering if you could maybe elaborate a little bit on how Philips and the academic centers? Because obviously the university is going to want the IP as well. So is this all handed over from the get-go, or how is this handled?
So it depends a little bit on, it's a little bit case by case. Of course, before we get to work with researchers, lawyers have to be involved first. So how it works typically is that instead of, we don't want to negotiate an agreement for every project. So we have what we call master agreements. So basically, we have such an agreement with University of Montreal, for instance. We have one also with the McGill University Health Center that will basically describe the different user case where how the IP gets handled. It's a little bit a case by case. If I can give general rules, and these are really general rules, I'm sure you can find many exceptions. Usually, all the previous IP, whether it's Philips or the institution, stays on that side, of course. Then if we have a shared project, then it will be shared depending on the percentage of contribution, basically. So basically, in many cases, what we will ask is to have the right to use the technology without having to pay. So basically, have license granted by the institution. But then if the institution wants to sell that technology to other people, we will also get maybe some money back depending on the percentage of our contribution. But it's really case by case. But what is always true is that it's always decided ahead of time. So, and that's the limitation of my work, is that if I want to start a project with someone, the first thing that I have to do is to make sure that there is an agreement in place. And that can unfortunately take quite a bit of time to put that in place because the lawyers from the two sides need to negotiate and agree on something. But that's always true that we have these agreements in place before we get to do the research.
So it's generally licensing, the mechanism that...
Most often it's licensing.
Okay.
Yeah, but again, there are exceptions to that rule. Yeah.
Thank you.
Yeah, I think the MR world, actually, if I can add a comment to that, the MR world has an interesting quirk in that there are also licensing agreements between the vendors. So GE, Philips, and Siemens have agreements to share, under some commercial agreements, to share IP. Because I think they're all realizing that they all have kind of a piece of a fairly big pie, and in a sense are able to maintain their control over that world in a sense. That's a quirk of the MR world, at least.
Yeah, if I can add, that's not something that is very well known, but that's true. So for Philips, for instance, if Siemens patents something, that's not so much of a problem because we have these agreements that we can share our IP. If then an institution that didn't work with any of the vendors did something and patented it, that's a case where we will have to purchase a license. So, but yeah, there are some license agreements already in place.
It's a bit of a reflection of how the MR world developed. There's a decent amount of openness in the MR world. Like raw data, you can get raw data off of MR scanners, for example, which is not necessarily the case for other medical devices where they're more black boxes.
Yeah, and I think in the end, basically, Philips owns enough IP that it would be impossible for Siemens or GE to do a system without using that IP, and the other way around as well. So basically, they found that the solution is that as long as we maintain basically our same level of patenting and getting new ideas out, then we can share. That's basically what happened.
I think there was another question here.
Well, I was just somewhat along the same line. I was wondering about your experience with the theft of intellectual property in the world we live in now. Data moves around a lot. Is that something you've had any experience with yourself? With Philips, that third parties get hold of your intellectual property and are able to market it themselves. Does that happen at all in the MR world? I mean, there's some countries that are well known for this, so...
Yeah, so basically in the MR world right now in North America, but also in Europe, there are basically only 3 players, Siemens, Philips, and GE. Then there are other companies, including Canon now, who used to be Toshiba, Hitachi, who I would say are quite well-established companies, but they have a very local market in Japan, for instance, in the case of these companies. And these companies, as far as I know, they respect the rules pretty well. Then you also have startup companies in China, especially. That's probably what you had in mind. And that's a little bit, as far as I know, I'm not at all an expert on that. It's a little bit more in the far west there. But yeah, most likely they don't respect our patents. As long as they stay on the Chinese market, I don't think we're doing anything about it right now, as far as I know. If they start to export things to either Europe or North America, that could become much more of a problem. But yeah, I'm pretty sure that happens, but I'm not at all an expert on that topic.
So in these two first world markets, if you want to call it that, how do the three companies distinguish themselves? Like, if I'm in the US, how do I know which one to buy, because they're all the same?
You should buy Philips.
Just buy Philips.
That's a good question.
Maybe you should ask me.
Yeah, so I'm probably not the right person to answer that one.
Just to answer that question, like if you look at the, in oncology, radiation oncology, the three companies, if you want to call it three companies, are very different from each other. It's like, we just look at the products, they are nothing like each other. So making a choice depends on what your preferences are.
Yeah.
But for MR, that's not the case.
No, it is.
It is. Sorry.
To some extent, the devil is in the details, in the implementation of the IP, and how well it's deployed. That has really distinguished different vendors.
Yeah.
It's hard to say one has really swept the board on everything, but there are certain areas which vendors are known for.
Yeah, so if I can just add to that, and again, I'm probably not the right person to comment on that, but for instance, I think Siemens, they quite nicely got a niche in neuroscience. They came out with a system with very strong gradients. That's not that useful for clinical use, but for neuroscience project, that's something that can be useful. So Siemens are seen by many as the neuroscience company. Philips focused a little bit more, I would say, on therapy. So one example is the MRI collaboration with Elekta. Philips work on IFU, so Philips work a little bit more on the therapy side than the other companies. So even though I would say for standard radiology work, all three vendors will do the work. So their system will do the work. For more specific applications, there are still some differences, I feel, between the
Companies have competed on price as well. One in particular, which doesn't need to be named in this case. And on the performance of the electronics underneath everything has also influenced the overall performance of systems, the reliability of systems, even over time. These are systems that tend to be installed for decades.
Yeah.
And so if they degrade quickly versus not, there's no moving part, there's a lot of heat, which tends to affect electronic circuits. And so there's things like that. And when sort of large sites make big purchases, they also tend to try and compete on multi-unit pricing, pairing it with non-MR. That's a big part of purchasing as well. So if it's a big site like this, and you're selling fluoroscopy systems and CT systems and ultrasound systems, if you bundle it all at once, it really broadens the competition aspect of it.
And it's very true for every modality, but basically, if you don't know modality, they will all look the same. But if you know, you will see quite a few differences. Yeah.
John?
Yeah, so I really enjoyed your presentation. Thank you very much. I mean, especially the general comment about how ideas get translated into products and the funnel pipeline and how prioritization works. So our experience with that kind of thing has been with small companies. The argument being that small companies have many less layers of approvals, and we've been kind of frustrated with large companies more on the radiotherapy side, that they're not moving anything. It's just, you have to convince so many layers of approval before they get interested in a certain piece of device or a piece of software. So in the case of Philips, where you make your agreements